as I was saying maybe earlier, that if I were to go through the whole program in detail, it would take certainly much more than 2 hours. So the objective is to introduce you to this this software tool and go through some of the more I would say important steps in more detail with some of the data sets that I have sent you. Now I will be not using the online version of it. I'll be using my personal one because it's much more faster, the response time. But those of you who want to just play around with it, please do click on the online version of it. And we are basically ready to go, right? So what I'm going to do is I'm going to share my screen. Yep, I think you can see it. Share that screen. Now, can you see the PowerPoint thingy in front of you?
Yes, we can.
Okay, so okay, so just at the very beginning, what I wanted to say is that as a plant breeder, the main objective is to develop cultivars, right? So sometimes people do make a big fuss about all these programs and they forget the main objective is to produce cultivars for farmers. So, DeltaGen, it is important for you to understand, it's just another tool that hopefully some of you will make use of it, and it'll help you enhance or, your breeding programs. As I said, another tool. So I call it a digital sort of tool, support tool for plant breeders, because obviously it's all to do, it's programmed in R and using a Shiny interface. And it was developed by Dr. Dongwen Liu, who's a colleague of mine. He did all the programming. I don't know anything about R programming. I'm more sort of my area is quantitative genetics and breeding. And Dongwen is I would say he is one of the best R programmers in New Zealand. I was very fortunate to work with him. So all the concepts of how different modules are linked to each other, it's all his works. Unfortunately, Dongwen can't be here today. Now, so you must be, all having this link. If not, you can get to the two papers that we have published on DeltaGen. You'll see the sort of references to those two papers at the end of this brief PowerPoint session before I get onto the actual program. And I just want to thank NAPB for inviting me to do this presentation. So let's get into it. Now, in today's sort of world of plant breeding, we have many challenges, right? And with global warming that all of us know, it's creating big problems around the world, and it's certainly having a massive impact in developing countries also. And in New Zealand, of course, I would say it's one of the leaders of the world in forage breeding. So DeltaGen started developing DeltaGen for forage breeding, and later on we sort of fine-tuned it to handle self-pollinating crops too. So when it was developed, it was very clear that forage breeders, like crop breeders, have a number of these challenges. So they have to be aware of that. This is some of the key challenges. Then we have breeders have many tools now, right? Phenotyping tool using high throughput phenotyping, molecular tools, based on genomic selection. You have transgenics, you have interspecific hybrid production. Then, of course, we have the toolbox of quantitative genetics, where we have experimental design, field techniques, quantitative genetic modeling, etc., etc., etc. So breeders have to be familiar with this, the tools, the challenges, and of course, they have to know, they have to have information about their plants they work with. So this is and as an example, we had to understand the traits and how these traits interact with each other, and try to use these tools to improve these important traits, understand the genetics that's driving these trait expression. Now, in New Zealand and in Australia too, we have the other challenge that plant breeders have. I'm sure even the tree breeders, for example, have these challenges. I'm not very sure about wheat breeding, etc. But but we at the moment have to breed for a forage value index. Now, that is, you have a combination of yields, persistence driven traits, like for example, white clover, we have vegetative persistence. So traits that drive vegetative persistence, and of course quality, and what will we look at in the future. Anyway, so we have to be able to develop selection indices. So all this makes it quite a challenge to plant breeders. So the question we asked ourselves is, how can we help breeders use this information to increase the rate of genetic gain based on cost-efficient breeding methods? Because you might have the best plant breeding tool in terms of achieving genetic gain, but at the end of the day, if it's too expensive, if the unit gain is too expensive, people are not going to use it. And especially in commercial companies, it's impossible to use it. So people are always trying to have cost-effective genetic gain. So to be able to do this, what we did was we thought we wanted to develop efficient and user-friendly decision support systems or tools that will enhance plant breeding. Now, it was important that these decision support tools were not complicated. They were simple to use. Now, at the beginning, people did ask me, oh yeah, but why developing another software tool to do analysis, and why can't we use SAS and GenStat? And of course, to me, those programs are the golden standard. However, unless you have quite a good understanding of quantitative genetics, etc., if you were to use some of the outputs like variance components, etc., coming out of those programs to be able to estimate genetic gain and look at the relative efficiency of rates of genetic gain across your different breeding strategies available. Say if you're looking at, for example, a cross-pollinating species, you might look at half-SIP family selection against full-SIP family selection, half-SIP family selection combined with genomic selection. So how are you going to do all this if you do not have an understanding of the actual quantitative genetics that runs this basically the breeder's equation? So to be able to use the outputs from programs like GenStat and SAS, etc., unless you understand it and also know some programming, it's going to be quite difficult. And often I used to get these questions from some of my colleagues who are extremely good field breeders coming up with really good cultivars, but they were finding it difficult to use these things, and especially with the new novel molecular tools coming on, how to integrate them into conventional breeding programs. So to do this, we started a program to develop decision support tools. So as I call it, breeders decision support tools. So we had a program, or it's almost sort of starting to continue the phase 2 of it in New Zealand, called pastoral genomics. And so I was responsible, it's not the word to use, I was responsible for running a stream, as we call it, to develop decision support tools for plant breeders. And so one of the tools was DeltaGen. So it's an empirical data analysis. It's basically, it's a deterministic simulation tool, and developed by Dongwen and I. And I think it was mentioned that my good mate Valerio did present a seminar on Eugene. So there was another group of us, and Valerio played a key role in developing QLine Plus, which is a component of QGene, developed by Mark Cooper and his team many years ago. So while DeltaGen is a tactical tool, QGene is a strategic tool based on stochastic simulation. And anyway, and one program could provide basic information to be fed into the other. And of course, the overall objective of developing these programs was to provide decision support to plant breeders working in commercial cultivar development in New Zealand. So now it has gone further than that. You know, DeltaGen has been polished up more to bring in the cross-pollinating species, some of the analysis, and it's been used across over 80 countries. And I'm not saying maybe it's not as, you know, it doesn't mean that it's so good, but I think it's freeware, so people use it. It's easier, you know, people have access to it, especially in developing countries. Now, so when you click on DeltaGen, you know, you get a, you know, the first screen. This is what you see. And as it was said before, it's freeware. We did not want to release it without having it basically looked at or commented upon by international sort of experts, and that's how we published 2 papers out of it. So if you look at the screen, DeltaGen, you see the you see the, you know, there's a tab here giving you the different components. I will go through that in more detail when I get into the program. And yeah, so the way it works is that it's very simple. There's data collected from the field. So basically, I'm sorry, what's this happening here? Yeah, you have the, now we also have introduced an experimental design, data entry spreadsheet, something like a data logger, which we are having a new version, the more user-friendly data logger being developed. Data comes in, you do your quality checks using plots and pivot tables, and there's an error, yes, you go back, you know, do your transformation, whatever, and if it's okay, then you get into the program. Just move these things onto another. Yeah, okay. Get right into the program, the analysis part of it. So you have linear mixed model analysis, and outputs from that can be also used for pattern analysis. That's a mixture of cluster and principal component analysis. Then it goes to multi-trait analysis. The feedback from that can be used in deterministic simulation, which also includes sort of a cost analysis, a very simple thing that will give you the cost per unit genetic gain. And then we have the Smith-Hazel index that could be used for multiple traits. And then you have reports being generated. So it's quite everything is tied up from experimental design, data analysis can go right through to the final sort of reports looking at rate of genetic gain, etc. So some of the main components, I would say the main components in DeltaGen, again, experimental design, quality assessment, linear mixed model, quantitative genetic analysis, prediction, then of genetic gain, breeding method cost efficiency, assessment, multi-location, trial diagnostics. That's a very important little tool. I'll show you that. And of course, you have the selection index stuff. It is also used in teaching. Quite a few places use this tool because when I developed it, I did not want DeltaGen to be a black box where you feed something in it and you get the results. And I think it's pretty dangerous. It's always good for people that use it to understand or know that, you know, what sort of theory is being used, what models are used. So there's when you click on help, so you can see all the, I'll go through some of those too and show you, but so we have all the main theory also there. And for students, you have references given and they can go and see it, etc., etc. Now, and as I said earlier, that there are 2 key publications one in crop science and the other this one in crop science and the latest one in scientific reports. This is quite an interesting paper. This is more introduction to DeltaGen. So for those of you who are interested, please do go ahead and read it. Now, so that basically ends the little PowerPoint presentation. Does anyone have any questions on what I have presented so far? Mr. Chairman, over to you.
Yeah, if anyone has any questions, like I said in the chat, feel free to post them in the Q&A and we'll try and answer them.
Okay, so let close that down for the moment. Right. So I think okay. What I'm going to do is to bear with me a minute. Put it down here.
Sophie, we have one question. They asked,
Yeah.
Is this software available for the public? And
sorry, can you repeat it? It's sort of you went off a bit. Is the software available for what? Sorry?
For the public.
Oh, yeah. Now, okay, so it's freely available. If you click on that link, right, you go to it and you can use it. But if any of you want to have your personal copy on your laptops or your tablets or what I can do is Villari or one of you can generate a Dropbox where I can put a zipped version of it, and then you just download it and run it on your program. So that has been very useful in some of the developing countries. We hand it over, we give the breeders that, and they're able to use it, without any problems at all. So yes, it is available, and that was one of the key reasons. One of the key reasons we developed DeltaGen. That's to help have it freely available for developing countries. I hope I answered your question.
Yes, I think
me?
yeah,
yep.
That answered it. So
okay.
They're
No
asking
problem.
for the link for the online one, which I'll try and post here real quick.
Okay. I should have sent you my presentation.
Yeah.
Do you want me to put it up again? Okay, hold it. I'll save you the thing of can you see that?
Yep.
Okay, so just bear with me that the link works really well. Only thing is in the last day or two, we have had a few problems that when you use the experimental design part, downloading the outputs can be a bit of a problem, but by the end of next week, all that will be working. All right. But as I said, if you want your personal copy of DeltaGen, I mean, we keep upgrading it, but it doesn't matter. You can use this, my version, the latest one, which is also, this is also the latest one. You can have it. So I'm happy to send it.
Sophie, could you possibly copy and paste that link into the chat?
Okay.
That's what they're asking for.
Okay. Now you might have to guide me through that. So where do I paste it? Go to chat, that one? Can you see the what I'm clicking on? Chat. So right. Where do I paste it? Oh, okay. So what I'm going to do is if I paste it there, did it work? Nope. Let me see. Give me 1 second. Just did it work? Right. So if I were to copy that.
So I put a link in the chat already, and it,
oh, did you? Thanks so much.
And so everyone was able to receive it. So
good. That's excellent. So what I'm going to do is I'm going to open. Now, can you all see the screen with DeltaGen? the program?
If you guys can see it, can you please paste in the chat?
Can you see my screen, DeltaGen?
We can see your PowerPoint.
Okay, so what I have to do is new share. So can you see it now, DeltaGen?
Yes, now we can see it.
Good. So when you are ready, I can start again.
I think I think you can probably start.
Okay.
Then
Okay. So about how many people do you know are there? Would it be over?
There are currently 19 attendees.
19. Okay, that should be good. Yeah, no problem. Sometimes when the number goes over about 50, I've had people sometimes 50. It just to get a bit clunky, the whole thing. The online version, because there are 90 people using it at the same time. 50 to 90. All right, so let's start. So when you click on the program, you have the introduction page with all the blah blah on it, and also it gives you the reference to the paper. Of course, we acknowledge the funders of Pastoral Genomics Plus, which was a consortia, using the different grazing industries in New Zealand, and also Dairy Australia. Yeah, so I need to acknowledge them for their valuable input. Now, so as you can see, it has trial design. Then we have data input. Then we have graphs and table. That's basically a quality sort of an check of your data. Then we have modules, and under modules, I'll go through these individually. Under modules, basically, if you under models, if you click, you have univariate, multivariate analysis, and then you have your pattern analysis, and then save and quick that. And also we have a help. It's a quick start guide. Now this, if you click on that, the quick start guide, it gives you a manual. I need to upgrade this. I'm in the process of upgrading it. But it sort of gives you a general set, of commands and bits and pieces of the program, which is enough to set you on your analysis pathway. Okay, so I must remind you that this was not professionally done. It might look a bit funny at times because I basically did sitting at my kitchen table. So it's a homemade program. Yeah, so it'll take you through. It's quite easy to follow, I think. All right, so it goes down. So you can have a look at it for those of you who have the link. So I think that is enough, and I will start with trial design. So under trial design, it is, as I'm not going to tell you, talk to you about trial designs. I'm sure most of you understand the importance of having an appropriate trial design. So we have completely randomized complete block, factorial designs, row and column, and p-rep designs, partially replicated designs. So find that most plant breeders that I know and I've spoken to, and in my lectures, etc., they use row column designs or p-rep designs, and of course there are randomized complete block design available if you want to use. So now under the design section, we have a default things if you are looking at complete randomized. So today, before, what I want to do is talk to you more on the row column design and something about the p-rep designs, which I think are quite efficient. So you can, when you get into this, you can write the name of your trial, and I just put some default options where the location is, the breeder's name, and then in this case you have default, you have 20 lines, and you have put in 3 reps. You can just change them, click and type in, do a bit of arithmetic and work out the number of rows number of columns. So in this case, we have 20. So 5 4s are 20. So each replicate will have 4 columns, 4 rows and 5 columns. So because we have 3 reps, the total number of rows in the design are 12. 3 4s are 12. And you have the number of checks. If you have checks, I'll show you that in a minute. If you have repeated checks also in your trial, you can have a vertical layout or a horizontal layout. You can cut and paste your entries, the names of entries, or if you can just leave that blank and it'll be numbers if you wish. And the random seed number. If you have 0, every time you press run, it gets a new randomization. Now, if you wish to have, to send a particular the details of your design to a colleague, or you want to have it recorded somewhere, you say, for example, if you have 23 12 5, that combination, and if you say put a seed number of, say, let's say 10, every time you press, you say run, you get the same randomization, right? So for the moment, let's keep it at 0 to have different randomization. So all you have to do is, if you have 10 here, the seed number, you get this will be the same randomization every time you press run. So let's have it as 0. And in this case, so let's run it. This particular example. So the program.
Zolfi,
yep.
So we send out that data that you sent us. Now, is that data something we can upload and follow on hands-on with you, or?
Yeah, you can. Just
Okay.
give me 1 sec for 1 sec. I just want to upload another version of this. It might be better. All right. So did I go out?
Yeah.
Oh, right. Okay. So what I'm going to do is, bear with me a minute. Recording. Share window closed. So what I'm going to do is share it again. Right. I'm sorry about that. Just that. How do I minimize this? For some reason, my connection dropped out a bit. So let me see. Just trying to see how I can. There. Right. I'm just doing a bit of a check here. That's fine. Fine. So if I'm just trying to get back to my right. Okay. Right. Sorry, guys. Just trying to get to this program here. Right. So let me open this. Right. And now I'm going to do is close this one. Copy up again. That's the right. So why can't I see DeltaGen? Why isn't it opening? Should be able to see it. You can't see DeltaGen at the moment, right? It's gone off.
You haven't shared your screen yet.
All right. So what I'm going to do is if I everything seems to have vanished. You can see, can you see me now?
No, you turned off your camera.
How is it? Okay.
Yes. Yep. It's good now.
Okay. I just can't, everything sort of, all right. Okay. Right. So what I'm going to do is if I share this thing, share, you can see me share the screen. You can see the screen now, right?
Yep, we see your desktop.
Okay, it's good. It's progress. Can you see DeltaGen now?
Yes, yes, we
Oh,
can.
great, great. I must apologize to all of you. Just that had a bit of a glitch here. Right. So let's go back to the trial design. We were looking at row column. And in this, so we are going to generate a row column trial design having 20 lines, 3 reps. Each rep you have 4 rows, 5 columns, and the total number of rows are 12. We are going to leave it as 0. We have not put any line names at the moment. So when we press run, so it's running a row column design. It has finished. Can you see the output?
Yes, we can.
Okay, so this is basically the spreadsheet that it generates. If you go to check design, you will see that there is a figure there, right? So giving you that randomization, right? So say you want to put in names, okay? Say you want to put in names, substitute this. So what you need to do is to, as an example, putting. So in that we have 20 lines. So these are just some names of cities in the UK. So you just copy them out. So we had 20 lines, just copy that.
Sophie, did you add one of those data tables
Yep.
as that data
Yep.
beforehand?
Yeah. It should be in your
you
data set.
would you be willing to show us how you upload that so you can we can follow
Oh,
along
just,
where you
yeah.
are right now?
So, okay. So just click on the spreadsheet that I sent you, having the names.
Okay.
So when you open that, you have names of towns. I just thought, okay, they're the entries. So you just, and I have just also on the side here, I just put numbers to guide me, in the demonstration, and you copy the just 20 names. They are the 20 names of your entries. Is it clear? And then you go back to DeltaGen. You go to here, enter line names. Just go there and paste it there. So they're in there now. And then you go and click run. Now it's running. And there you are.
So I'm looking at the online version right now, and there's no way to generate that data to get to where you're at. There, like some of those are just missing for me online.
So did you go to that? Maybe the glitch I'm talking about in the design thing that we have had.
So it literally only has the design type, random seed, and that's it. So.
Oh, so just try opening it out again. Just go out of that and click again. Get out of click the link again.
Yeah, it's the same. I don't.
So when you just generate the design without adding any entry names and click run, does it give you the design check after it runs?
It doesn't even there's no place for even the name of experiment, location, place, name of breeder, number of lines, rows, any of that on this online version. Oh, but some people are able to do it, it looks like, according to the chat. So
okay.
Is anybody else able to see those and follow along right now? Okay, some people are not, some are. does it work on certain browsers? Is that maybe? Or
Could be. I have to ask. All right. So what the best thing I can do at the moment is when we finish the demonstration, right? If one of you could send me a Dropbox where I can copy the zipped file and send it to you, and then you'll be able to have it on your. The best thing is to have it on your personal computer, because
okay.
you know, sometimes when you go onto the field, you will see that you can even, you know, do your analysis sitting on your field when you collect the data immediately just to check. So for that, sometimes you might not have internet coverage. So if you have it on your computer, on your tablet, you're fine. You're fully independent. Okay, I mean, so, all right. Sorry about the design thing, but I'll just keep going. Then you will know what you can do with it. Okay. So say, for example, we want to include some checks, repeated checks in this thing, in this design. So say we have 20 lines, actual lines, and we want to include say 2 commercial checks, right? Check 1, check 2, may call it or whatever, give it a name. So therefore, you know that you will have a total of 22, right? 20 of your entries plus 2 commercial checks. Let's say you want to, because of the commercial checks, because the repeated checks, you can reduce the numbers, say, to 2 replicates. And each commercial check, say you have number of check positions in replicates. So each commercial check has 5 repeats in it. So 5, you can click on the default, let's say 5, 5. So therefore, the total number, since you have 2, the total number, because of 5, each one 5, the total number of individual lines or the plots in each rep will be 30, right? 20 plus 2, each of those 2 checks are repeated 5 times, so you'll have 30. So in this case, you can have 5 6s are 30, and you have 2 reps. Each rep we have 6 rows, right? So let's run it without names at the moment. So if you run that, I just want you to see how the analysis comes out. So we can just run it random seed. Let's say we can just run it, and you'll get numbers, not your entries. So there you are. So these are the rep 1, rep 2. So these are your rep 1, your 2 reps. And these are your commercial checks, or whatever you want repeated, right? So if you think that the distribution of your checks, you don't like it much, you just press it again, and it will re-randomize everything because the random seed is 0. So it re-randomizes it. So now you want to put in the checks with the names of your entries. So then you can prepare a spreadsheet, or you may have that somewhere. So in this case, so here I have prepared it already as a demonstration. So always your 2 checks are the first 2, right? 2, 3, 4, 5, whatever number of checks, but the checks have to be on the top, the first rows, right? So in this case, we have 2 checks. So we have row 1, row 2. So basically go down, and then it copies all that. We have 22, right? Altogether, 22 entries. Right click, copy that. Go back to your program, and it's asking you, please enter 22 line names. So that's what I'm exactly doing here. It tells you that. Paste. And now we run. If you wanted the same randomization, of course, we should have given it a number, random seed number. We have a 0, so it'll give you a different randomization. Oh, why is that? It should be checked. So please enter 22 line names. That means I have not entered the 22 correctly. Why is that? So let me see. check 1, check 2. '21, '22, maybe I had 2021. Copy. Let's go here. Paste. No, for some reason, this is not working. You should have got the check numbers here. Does it work for you? Doesn't seem to be working here. Did you try that, Kevin?
In the chat, someone said it may be because you did not delete. Please enter 22 line names at the end of when you enter the names. That was
Sorry,
somebody in the
did
chat. If you look at that, somebody said maybe it's because when you entered it, you didn't delete the
could be,
line names.
could be. I haven't done this for some time. Let me see. So let me copy it again and let me go here. I know what's happening. So what I'm going to do is I'm going to refresh it. This is happening. So right. So now we go to trial design, row column, and right. So we had 22 lines, 2 replicates, and enter names here, number of checks, 5, yeah, and we enter the names here. Paste. Now it should work. Yeah, it's working now. Too much of information. Yeah, there you are. Okay. So we were doing, I was trying to do too many things at the same time. So there you are. So you get your check positions and you get your that. Okay. Now we have that and we can save the result. So if you want that to be transferred to a spreadsheet and to save on some database, it comes as a CSV file. So that's your thing here. Now you might ask me why you have the check separately, because when you do the analysis, if you're doing some like a genetic analysis, you don't want to confound your actual additive variance or genotypic variance with the checks in there, right? So you can use remove the checks as fixed effects. But anyway, that's another day. And also I'm just telling you, it gives you the spreadsheet, right? Where you can and save it as an Excel file. Now, so if you go back to that, you can also save your layout. The layout comes out here as a CSV file. So that's the thing. So there you are. So this again, I have to tell you, there is a problem with the program because I told you about the software glitch which we're working on. But if it's a row column design without checks, you get the whole thing there, right? All the numbers. I'll send you the original version, then all that will be cleared out. So go back here. Now you can download this picture. Either you can use your snip tool and just cut and paste, or you can just go to a Word document as a Word document, click, and it will download Word document. There you are. So it gives you a report. If you go down that, there you are. You have that, right? It's a pretty handy thing. Gives you a nice, right? So that gives you that. Now let me just close that one down.
Sophie, we had one question on if the program allows augmented as well.
So the row column design is an augmented type of augmented design, right? So they're augmented designs, right? So am I clear what I'm saying? Or so these are augmented designs.
Yeah. Although, would you be able to, you post that question in the Q&A, would you be able to post in the chat if that clears up your answer?
Or send me an email. Send me an email and we can,
Okay.
I can answer more on that. All right. So we have generated our design. So if we go to now design results, and if you go towards the bottom, you will see that we have an option called serpentine. You'll see in a minute why what I'm talking about. Start at the top corner, top left, top right. So you can basically, this is to collect data, right? The direction, horizontal or vertical. And it also gives you options of traits. So if you click on a trait, yield, growth, plant height, you can also, or if you don't like this, if your trait is AB or something, trait AB, all you do is try AB and add, right? So let's say the trait is AB. So now what is all this about? It is about generating your, if you want, labels for your seed packets and your planting order. So when I say serpentine, it's that way, right? So starting on the top left, starting on the top right, that way, right? So that is horizontal and vertical, starting that way, top left, top right, right? You can basically print out a spreadsheet to collect data. So basically, you generate your design, you save your design. You can do it on the field if you want, and you have done it before you go to collect your data. And if you click this after putting up, say we want to start on the left and go zigzagging. And if you click send to input, it takes you to a spreadsheet, right? It takes you to a spreadsheet. And when we come here, what we have to do is sorry, I'm just playing around with this. When we come here, what we have to do is click on view data so that it's go to this one. We have to update so that it's then loaded into the program, right? Sitting on the surface, update, and go to data input. So there you are. So this is your, so you have check 1, check 2 if you want. This you can, and then this is where you go. So if you look at the columns, the way you go, if you go zigzagging, 1 2 3 4 5 5 4 3 2 1 1 2 3 4 5 5 4 2 3 2 1. So it's you can start to enter your data here, right? So it's just a spreadsheet. Say you're on the field, you're entering data 12. So let's add some data stuff here, some dummy stuff here. Something like that, right? So just to cheat the system, let's fill it up. So you have got the data in now. Say you're on the field, you copied your data. Then you go down, and then always update your data, right? So that your latest version is there from time to time as you're collecting your data. So your data is in there, and then you can download your data, right? And of course, this gives you, of course, remember that the breeder's name, all that you have initially entered, the year, the season, in this case, perennial species, but in your case, it may be not season, you could call it something else, but you know what it is, the location you have collected, and your replicates, right? Replicate 1, 2. So this is basically your experimental design, right? So now what you could do is you have updated the data, so it has gone into the program, and then you can download your your data sheet as a CSV file. If you download it, you get it all there. So there you are. So that's your data. Okay, and you can save it in a database or whatever place you want to save it in. Let me see if I take that. Do you want to don't save? Clear this up a bit. Right. So now the other thing to remember is that once you collect this data from the fields, you have this data, you might want to check the quality of the data. So to check the quality of the data, what you do is you go here. Now, before I proceed, am I clear? Have you sort of got an idea of what I've been saying? And is it clear?
It's clear in my end. If anyone has any questions, here. I think we're good to go.
Okay, silence. That's good.
Yep.
So if you want to check the quality of the data, do a very quick check. You just go here and you can click on plot. And in plot, you have sort of depending on the type of data that you have. So I generally use a histogram and density plot. And I want to check the trait AB. And it's sort of remember that this is mock data. So it'll give you a distribution. We'll have a look at that with some really data. And then if you want to look at it by replicate, you can click replicate. So replicate 1, replicate 2. All right. Then there is also another way of looking at your data. The actual with regards to the experimental design, where the actual plots are. This is very helpful. You can go to your pivot table. You call up a heat map. And here looking at averages because you're looking at every plot, or maybe it's a single plant, sort of just one value. And trait AB, that's the data. Because you might have multiple traits measured at the same time. So this, we have one trait, AB. And you have replicates. Put the replicates there. You can have your rows, your row data comes on. And you have your columns. So this gives you a heat map giving you your lowest values, sort of your intermediate, and your highest values, high values. So if you're, checking, you have done some measurements in the field. Often what happens is we go to the field, do our measurements, we take the data, we save it on a database. Then a couple of months after that, we have the data and say, wow, this is funny. This particular data point is not right. But really, it's too late because in annual species, you may have harvested the crop by then. In a perennial species, it's a different point in time. The plant has continued growing, it has changed. Its morphology, the phenotype has changed. So it's always, this gives you the opportunity of having a look at your data on the field before you leave it, this heat map, and saying, well, this particular value is not right. There's something wrong. So I would actually walk up there to the plant or to the plot and say, maybe I have missed a decimal place, or I've hit the wrong key while I was entering data. And then you can go back to the data set and fix that point. Okay, correct that point. So this is really helpful because of that. So let's go to trial design quickly and have a look at P-rep design. So in P-rep designs, basically the philosophy of a P-rep design is that you have, say, about say 100 entries, and of that 100, say maybe 80 of them might be, 80 of them you will have only one replication, and another 20 of them you might be able to replicate more than once, right? Partially replicated designs. And so in this case, in this default, the example I've given here, there are 80 individuals. Altogether there's 90, but 80 of them have only one. You can afford to replicate only one, right? Because the maybe just a small number of seed. Here, 10 of them we can replicate twice. Each of them can be replicated twice. Okay, so when you look at that, the total is 100, right? Twice, so 80 plus 20, each one replicated twice. So number of rows, number of columns, you have 10 rows, 10 columns is 100. Then we have a block sequence. So what we thought of also, just if you can balance it out, you can have some sort of replication in the overall design. Okay, I'll show you what I mean. So similarly to the row column design, we have a random seed number. You can, have the same design generated, or you can have a 0 here, which every time you press run, it generates a different randomization. So in this, let's generate the default one here. So it runs and then it's generated. Let's look at the design. So there you are. That's your come on, this is how to get rid of this. So here you have your 10 rows, 1, 2, 3, 4, 5, 1, 2, 3, 4, 5. And you have your 10 columns, 1 to 10. Now, each of these is replicated twice. So if you draw a line, count 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and draw a line. This will be sort of the halfway point. Replicate 1, replicate 2. So the way it separates these things out is that on one side of the line, you get 1 rep of each of these lines that are replicated twice. And on the other side, rep 2, you get its second rep. For example, 84 here, you have 84 on the other side of the middle line, right? 82 will be somewhere on the other side, 82 here, right? 90 will be here. Okay, so you can, let's do another, just one more example of that. So let's do, I have one example. I'm just thinking of an example which might be appropriate. Bit of arithmetic, I have it written down here. Say, for example, we have 100 individuals with only 1 rep. Then we have 10 individuals where we can have 3 reps. And we have another 10 individuals where we have 2 reps. Sorry, another 10 individuals we have, sorry, but so 3, I had to put 3, and this one 2 reps. Right, so the experimental design, the total is 150 if you do the math. So we can have, say, 15 rows, and we can have 10 columns, leave them as they are. We can continue with, so we can basically have something like a 3 rep result. So each of the reps, these are false reps, but each of the reps have 5 rows and of course 10 columns. So you'll see how it works in a 3, 4.
Selfie, we're kind of losing you. Oh no. I'll try and get him back on.
Can you see me and hear me now?
Yes, yes, we can see you.
Yeah. Well, okay. So let me have a look at this here. What's happened? What's happened? Okay.
Selfie, we just had one question on
Sure. Please do.
on how you deal with missing data when you're importing your own data.
Oh, okay. I will come to that in a second.
Okay.
I will come to that. I just want so let me keep going further. So so this is an example of partially replicated designs, right? So it's a similar to the other designs. You can download them as CSV and you can download it as a Word document, right? I don't want to repeat myself. So let's go now. So let's go. So you have an idea of the design part of it. Now let's move on to what I want to do is refresh. And let's go into data input, right? This is the interesting part, like. Now, in data input, we have upload, the standard uploads. If you click on browse, you can upload data sets. So you have these data sets, which I've sent you. So if you, for example, upload the CSV, wait, let me, was this the one? Oh, I'm just hoping that we'll be using the same data. No, let me documents. I want to upload the same data that you have. ABC Canada. All right. So I think this is the material that I sent you.
Yep.
So to be able to upload your data, the data should be saved as a CSV file, or it should be saved as R data. Okay, if you're uploading using the upload option. Okay, now let's get back to this one. Now, before you upload the data, this is very, very important. Now, I think I'm going to answer the question that was asked. You have to define, if you have missing values in your data set, you have to define them. You have to tell the program how you have saved them, entered them in your data set. So if there are no missing values, leave it as empty. However, if you have missing values, the missing values can be a dot or an asterisk or empty. Okay, so to be on the safe side, what I do is I just have all my missing values as empty, so it just uploads the data without any problem. Okay, so in this case, we want to upload a file. So in this case, I know that there are no missing values in this data set. So I go here, data entry format, matrix, this single site data, click on that and upload. Here's your data. Now this is a row column design. Put it at the bottom here. Keep it there. Right. Come on. Keep it up there. So this is a row column design. Now the data is not in your software yet, in your program. It's just sitting on the surface. Right. So what you do now is you have to make sure that the program identifies or detects the particular names you have given. You have named your data, your columns with. So some people might call a rep a block, right? So then what you have to do is you have to click on reps. We don't have years, seasons, locations, nothing like that. So in this case, we have it as reps, but if you had it as blocks, there'll be blocks here. So you click that, and there it's taken it up. Row, column, and you line. So in this data set, we have rows and we have columns. So we have rows, we have columns. We don't have samples, we don't have checks, but we have to say lines. The lines are there, right? And then you click. So now you have defined your columns, what your columns are, match them up with what the program looks for, and click run.
So what would be the difference between sample and line?
Okay, so when you collect, say for example, you have a plot, right? A large plot. You're a wheat breeder, so you have a large plot, and you can't afford to harvest your whole plot, right? To get your yield. So you might, because it's too large, so you might go and take 4 random samples. So each of those random samples will have a value. So what you could do is you might add them up and divide them by 4, which you get a arithmetical mean. However, around each of the samples, you'll have sampling error. So when you run your linear mixed model analysis, you can remove you can partition out the variation due to sampling. All right. So if you had samples, you'll have another column in your data set. You'll have another column in your data set called samples. You'll have, if you took 4 samples, you'll have 1, 1, 1, 1 so you'll have sample 1, sample 2, sample 3. So you're telling so when you put samples, you're informing the program that there is another variable called sample, a factor called sample. Did I answer your question, Kevin?
Yep.
Okay. And then you have your checks. So now we have pressed run. And so you can see these. So these are all factors, and line is it's your variable here, which you're going to measure, or your trait. And if you, so it gives you a summary of that. Then you can also look at things like structure. You can summary. All these programs have this. It's it's median and the mean and the quartiles and all that stuff anyway, right? So you can play around with that. So let's go. So now it's in the program. You can then have a look at it. Again, remember that this is a mock data set, right? You want to have a look at your data histogram. You want to look at yield. So there you are. It plots your yield, and you can have a quick look about its distribution. If you want to look at its distribution by replicate, click on rep, and it gives you rep 1, rep 2, rep 3. And if you have, reps within so many locations, you can partition that out. All will be shown here. Okay. If you want to change it to a density plot, you just click on density and you get your density plot. So you have to tell it you want yield, and you want, yeah, there you are, things. So you can do a few bits and pieces. Now you get to models. So now we want to do, because it's a univariate analysis. And so let's assume that this is the default is half SIP families, then you have full SIP families, options, and you have self-pollinating crops, say for wheat. Then you have early generation trials, right? Early generation trials generally are, they run them using large numbers. You might use a P-rep design, for example, okay, non-replicated. So yeah, so the default we have put in is half SIP families. And it can be there. It depends on how we interpret it. Interpret the results, the variance components, if you run a random effects model. So the primary trait is yield. So in this case, there are no fixed effects. So you click null, and then your random replicates. This is nested, rows within replicates, columns within replicate, line, and if you want, you can put your line by. However, what I want to tell you is that this is very flexible. We have given the breeders options, but if you do not want to have if you want to have just rows and columns, all you have to do is delete it and then type what you want. Say, for example, you want to analyze this as rows. You can just type rows here, ROWS, and add rows, and it adds it in, right? So it's very, very flexible. Okay, so let's just put them in here just for the moment. Where is that? No, let's delete all this. Right, let's start again. So you have replicates, rows, columns, lines. That's what you want to have a look at. Now, if these were just genotypes, you may estimate a broad sense heritability. If they were genetic families, say if they were actual half-zip families, you can estimate narrow sense heritability on a line mean basis. And then you want blobs because your lines are random lines. Then what you do is, you can also have a secondary trait. If you want, you look at things like correlated response to selection and things in the later on, but let's keep that at the moment simple. And then you hit run. So it's done. So it's done a RAML. The RAML analysis is here. So it looks at your variance components and standard errors for your bits and pieces in your linear model. And we also have a p-value, a log likelihood ratio, which we have used, and it gives you a p. So it's saying that highly significant, not significant, not significant, not significant. Remember, this is just a dummy data set, and it is giving you a line mean heritability of a breeder's dream of 90%, right? If it was half-zip family, you say that, it's 90% additive variation, right? Everything you're looking at is, is almost additive. So, I mean, this is false, right? It's a dummy data set. And then you can go in here, and if you click blob, it gives you the mean of the population of individuals that you have looked at, right? So you have 42 individuals in this data set, if they're half-zip families. So this is the mean of all those 12 half-zip, the blob mean. This is your error coefficient, correlation error, and then you have your blobs here, your random effects, and you can sort. If you want to sort from the highest to the lowest, just click sort from the highest to the lowest. You have the standard errors, lines, and if you want to, obviously you want to print this out or you want to save it, right click, download CSV file, open. There you are. So you got everything there. Your analysis is done with your standard errors and your lines, right? And if you had line names, all those names will come out there in your trial, in your entries. Okay. Now let's don't save. Yeah. Okay. So go back to this. Also, if you go back to your models, your basic model, and go back to the top here, you can look at your residual plots to see whether it's normally distributed or not. If you click on that, it gives you, right, an idea. So this is sort of all right, I would say. Well, if there are biometricians in your crowd, you might disagree with me. But all right, so we have looked at it. And then if you want to, if you wish to transform the data before you analyze, you just click on that and run your analysis. So it's there, and then you analyze your data again, run to look at your log transform data. So that gives you different pictures. So I always tell my students, be very careful when you're transforming data. If it's slightly skewed or anything, it's fine. Analysis of variance is a very, very powerful tool. If the data is really skewed, I run to the biometrician to help me, or you might wish to not use the data, because I always tell people that when you transform data, you're really not looking at your actual data. If there's a big problem, just drop it. Don't use your data. Otherwise, use the appropriate transformation if it's slightly skewed, if it's, well, skewed, but not really, skewed too much. So there was something about transforming data transformation. So let's refresh it. Do you have any questions? I'm going to a more interesting data set now. So let's go to clipboard. I'm going to do it with a clipboard now. So I'm going to open and bring in a data set with where it's asterisks across sites. I'm going to take that data set. I'm going to open that data set now. It's sort of a, a part of a larger data set, but you have it in, this is pretty cool, sort of a more of a larger data set. But here in forages, we have years, we have seasons, we have locations, replicates, rows, columns, lines, and growth. Now here, you see that there's a missing value with an asterisk. Sorry, go on. Yep.
You're still showing the gen right now. Are you like the actual program?
Has gone off, has it?
No, it's on. Are you looking at another data set right now?
I am looking at a data set. You can't see it, can you?
No, we can't see it.
Oh, sorry. Thanks for, I'm just get carried away with my program sometimes, and I forget that people are listening to me. Yeah, okay. So what I'm going to do is stop sharing. And now I'll share. Oh, where's that? Is it that one? No, it's this one. Can you see it now?
Yes, we can.
Okay, sorry about that, guys. This is a data set collected across several years, seasons. So this is perennial ryegrass, and we have 5 seasons: early spring, late spring, summer, autumn, and winter. So spring, early. This is at some sites in New Zealand. Short. This is a site called Kerry. Replicates, rows, columns, lines, and this is growth score data. Missing value here has been defined as an asterisk. Okay, so I don't take sort. I always have sort in my data sets for obvious reasons. So I just don't want to take that into the analysis. So I copy it, copy the data. Oh, what did I do? Come on, Sophie. Right, let me go back. Right. Oh, what am I doing? Right. Copy the data. And now I'll stop sharing. I'll go back into DeltaGen. I think it's here. Share. Yeah, so I hope this is the one I was using. This one. Yeah. So data input, I'm using a clipboard. Now I told you that before you upload your data or copy and paste it, you have to define your missing value. It's an asterisk. You can all see DeltaGen now, right?
Yes, we can.
Now I paste the data in here. And I submit the data. So the data is on the surface of the program. Now I have to define these columns, right? So I go down to the options. And we have years. we have seasons, we have locations, replicates, we have rows and columns, we don't have samples, we don't have checks. I don't think we have checks, no. And we have lines, could be anything, half sib, full sib, self lines, whatever, right? And then we enter. Now, this tells us that the data has been gone in successfully. NA means a missing value. Say you can say 53 NA. So it has identified it as a missing value. It's telling you that. And then you can go and look at the structure, etc. how I showed you. So now let's go into the models. And if you wish, you can go and check the how the data looks like, etc. Go into your univariate analysis. Our primary trait is growth. Now, again, it depends on how you look at fixed terms and random terms. So in this case, 2 years, 5 seasons, 3 locations. Now it depends on what you take as random, what you take as fixed and random. Okay, it's up to you. So I will just put these as say random, year, season, location as random, and so here this is columns within replicates within locations within season. Then you'll have column within replicates within season and rows within replicates within season. So nested. So rows, and then you have columns, then you have your lines, and then these are your random effects. And then you can have your things like line by location interaction, right? Line by season. So to make it the analysis faster, let's put it as line by location. You can have your heritability, what type, depends on the data you're using, your lines, or you can also have, I'll just run the analysis and show you something else in a minute. So now it's running the linear model. It's estimating the variance components now. And there you are, let's run it. It would have taken a bit longer if I had the line year, line season interaction. So in this case, it's telling you that these are your random effects, right? So it has done an F test, and this is the F ratio value. So I would say this is significant, this is not significant, and the locations are highly significant. So, right, so this is the fixed effects part of it, and the random effects output, you will have it here, right? Line, the variation, and your interactions, and so it has done a log ratio, log likelihood ratio test, and it's saying that very significant, highly significant variation among your the interact here. So that means your rows, down your rows, high across your rows, high variation, columns within replicates, within location, the season, within year, high variation, line by location, genotype by environment interaction, highly significant, line, significant, and then your replicates within, yeah, it's also significant. So it's giving you that, and it's giving you a line mean heritability for growth of 0.77 or 77% with the appropriate standard error. Now you can go in and look at your blobs here of them. Again, as I showed you, you all have your blobs. You can, from the line 1 downwards, you can sort your blobs by from the highest to the lowest, going down, and you can download it and use it. Now, say for example, So in this case, you see that you have three locations. You want to have a very quick look of how those lines performed across locations and how those locations, what's the relationship. Just a very, very quick look until you get to a more detailed analysis. So if you were to, what you do here is before you run the analysis, you go to modeling your results, and you want to generate something like a pattern analysis. So you tell the program, yep, I want to generate pattern analysis, but for that, I need the blobs of each line at each location. So I put line location blobs, right? And then I run the analysis. So you get the same result, but you'll get a blob of every line within each location. So it's calculating your variance, estimating your variance components. And now if you look at your blob table, right, here you are. So there are three locations: Kerikeri, Palmerston North, and Ruakura. These are three locations in New Zealand, important locations where we test our material. So for line 3, the blob of line 3 is given at each of those locations, right? So similarly, you get for each of them. Then what you do is you move this down. You go up here and click on pattern analysis cluster. So it has clustered it already, taken the data and clustered it, and given you a relationship, a dendrogram showing the relationship among those three locations. Some useful information, you know, to look at, to consider. And of course, this is the primary direction, and the secondary it has looked at all the lines separated out into all those lines into their particular groups based on their performance across the three locations. And here the locations are grouped based on the performance of those lines, right? This is all growth, of course. Then if you were to click on that, it gives you, I would say, sort of a biplot, okay? And then it starts to say that, you know, and I'm sure most of you know how to read biplots. So this particular group 1 is showing some potential for specifically these lines for broad adaptation, I would say, across these three locations. And these locations, Palmerston North and Ruakura, are closely have a higher correlation, positive correlation than, say, for example, Kerikeri. Okay, and you have site-specific individuals if you are interested in that, and then you have a particular group here, group 3, which is performing below the mean of this whole population of lines. So that gives you a very quick picture, graphical summary of what your data is saying. Do you have any questions about it? And then you can do your residual plots if you wish to see. They're pretty good. These are pretty good, I think. So you can look at that. And does anybody have any questions on that?
Please post any questions you guys have in the chat.
Okay, silence.
Yep.
So if you click on help, help opens out basically the theoretical information. So this is the general equation for your heritability on a line mean basis, right? This is for broad sense heritability. Then you have your narrow sense heritability here and all the definitions. There's also genetic correlation because we haven't done correlated response to selection. More, and there are some important, well, interesting, useful, I'll put it that way, references that you can use to look at the more theory if you're interested, if that is what you're interested. You have definition, very crude definition of what a blob, how the blob is calculated, and blues, how they're worked out, right? So you have that again, if you're interested. I find it, you know, very useful when I'm working with students to explain to them practically how you can get data and how it's actually analyzed. You can, if you want to now, we can do something else by deleting these two and putting lines up here as fixed effects and see the difference. So you can get a line by location, interaction, line by location. Where is that? Line, location, fixed effects, right? So obviously you can't get a hedgeability for fixed effects because there's no variance component estimated. You obviously can't do blobs because it's fixed effects, but you can do blues, lines, and we can run that and see what happens. So it's running your mixed linear model. So this is the time to ask me any quick questions if you have. Hopefully you have understood. And look, it's a matter of taking the program, working with data sets and learning. You know, the more you do, the better you get at it. It'll do the last minute estimates. Kevin, sorry, go on. Yeah.
We have a question on if you can perform tests or any kind of pairwise comparison tests.
No, we don't do pairwise comparison tests here. It is important to understand that. Okay, let's talk about the blobs, right? The blobs, they're basically giving you the genetic worth or genotypic worth of the individual, right? So when I do select material using blobs, I sort of look at compare the individuals based on their standard errors. Whereas when you do when you put lines as fixed, like what we did, and when you click on blues, because we wanted blues, this is what you get, calculating blues. Then what you do for that, you get a standard error of differences, you get your LSD, least significant differences, and of course here, I use the LSD, either the minimum, average, or maximum, depending on the accuracy to which you want to go, to make differences, to pick the best out or the worst, depending on what way you want to select. So here again, you can, these are your blue standard errors. Again, you can sort by line, you can sort by blue, and each one has its standard error, and then you have the LSDs. Did I answer your question?
Yes, I believe so.
Okay.
Is there any limit on line numbers or number of environments or combinations?
No, there's no limit. You can go. It might take a bit longer to run, but yeah, it also depends on how fast your computer is. So I'll show. Okay, so let's do a very quick run to the genetic analysis part. I know that's basically it's another talk on how to perform your genetic analysis. So let's go here. Now, for example, let's assume that, let me go and take lines out of that, line by location interaction out of that, and get down to, let's put them back into the random model line. Line by location, obviously, you know, if I were doing it, I would use line by location interaction, line by season interaction, line by year interaction, and then you have the three-way interactions, etc. Depends on how much you want to partition out your data. So in this case, we are up, that's all right. Let's calculate. Let's do an estimate of that, and we want to line. And so what we have selected is your let's do half seed family. For example, I could have done in red lines too, but it doesn't matter at this point in time. Run the analysis. We'll give you an idea. I just want to give you an idea of what you can do once you get a good data set. And if you know the structure of your population, whether they're half seed families or not. Right, so what happens here? Modeling, so this is got, and you can have blobs here. So this is what we did that time earlier on. So you have all your variance components, your heritabilities, etc. Now we want to do, say they're half seed families. We want to do an analysis, a genetic analysis using some of the breeding strategies, breeding equations available for half seed family selection. So you click on that and it takes you to a different screen where it gives you different selection methods. Half seed families as it is, with progeny testing among and within half seed families, genomic selection straight up, half seed family selection, correlated response to selection anyway. This is among family phenotypic selection within half seed family, genomic selection, among family and within family genomic selection, right? So you have a whole lot of individuals there. So here, standard, industry standard. Now, what is the industry standard? If you have a check that you want to compare with, right? So for example, if you go back to modeling, if you go on to blue, say you have a check, a cultivar. So this is the mean of all those half seed families. You had 86 half seed families, the mean. Say you had a check which had a value of say 5.2 or something like that, right? So that is what you put in here. You put 5.2. That's the actual mean, blob mean, 5 to 5.2. Say for example. Then we want to start with that. Your selection pressure among your half seed families. You can have different selection pressures. So let's start with 20% selection of your top. So you have, in your trial, you have 2 years, 5 seasons, 3 locations, 2 reps, 1 sample. Then you want to put some costs. So you can find you had 86 lines. Say if you're to take 1 sample, if your cost was, say, let's say $1 as an example. So for each particular replicate, if your replicate it costs, say, I don't know, let's say $600 a replicate per year to run. Location trial, say 25,000. General costs for crossing and whatever you want, you can make it if you want to. You might say it's general things about, say not 20, say $1,000, 1, 2, 3. Or you can leave it blank if you wish, right? It's how you want to work it out. Okay, so you update and it's run the analysis. So what it's saying, if you put a 20% selection pressure, your genetic gain in absolute units will be 0.39. 7, say you get a 6.89% increase. That is, you get an increase, your response to selection will be 6.89 relative to the mean of the original population. So what does that mean? Mean of the original population, what it's saying is relative to, go to models, go to your blobs relative to that. But that's the mean of your 84, right? 86 lines. So relative to that, you are increasing, you get an increase of 0.3997. Remember, it's a scope that they were scored, and related to that, the increase was 6.98. Now, this is relative to the so I didn't change this one. So we said industry standard was 5.2, I think I said. So if your industry standard was 5.2, you have your improvement is 18.42% better than your check variety, okay, or industry standard. So this is the gain per year. This is the cost for one cycle of selection. So in this case, the cycle of selection will be 2 years field testing. We also add another one in cross-pollinating species, another year. So it's basically 3 years per cycle. It costs that. And then this is, I can't remember now what that is. Cost percentage. So this is the cost per percentage gain. So to get every percent, you spend $23,000, $23,712 for every percent genetic gain. This is the cost per year. And this is the standardized genetic gain that is divided by, so that is the absolute divided by a cycle divided by the phenotypic standard deviation of the trial. Okay. So if you want to say, so that's applying a 2%, 20%, you can look at a 1%, right? And then you can update that. So it gives you, it shows you, sorry, it shows you, the increases, as you start to increase the selection pressure, you're getting, obviously, it's obvious, right? And looking at costs, obviously your cost per cycle of selection will decrease, right? Because you're getting a higher genetic gain. So per year, and this one cost per cycle, yeah, it will decrease 163,000. Yeah, so it's decreasing or increasing? Oh, this one here. Right, so that is relative to, I always forget that. Again, yeah. Yeah, so 163,000. Where is this? What's happening here? Yeah, this has increased. That's correct. So you get the appropriate costs, and of course you get your efficiency of your selection is starting to increase, right? The standardized from 0.554 to 0.68. Then you can also say if you want to compare this with genomic selection, so it goes into looking at selection within family, the accuracy, and there's room for among family, within family, and so you can also do simulations like right. You can say, what if I ran my trial with only 1 year data, or you might say you had only 1 year of data, and you had only one replicate, and you have only one location. What would be the result, right? What will be the overall result? So you can see that, you're getting less genetic gain for obvious reasons, right? And so you can do all sorts of simulations like that and demonstrate to students the importance of having replication, right? So one replicate means there's no replication, right? And at one location, just 1 year across 5 seasons. So you might say that anyway, we don't want to get into that. There are a lot of things you can do, a whole heap of things. And then if you go to help, help gives you all the different readers equations that you are using, that you can use. Full SIB hub, this is for full SIBs, and all the definitions there. We also have correlated response to selection. I just don't have time to show you all that, I'm sorry. Then equation used for inbred lines, right? So here, this asterisk here, for example, is obviously if you're familiar with that, for inbred lines, the genotypic variation among your lines will vary from your generation, right? Or your selfing generation. So you start with a selfing 0 of 1, where you have your coefficient of inbreeding is equal to 0. But as you start to self, that changes, right? As the lines start to get fixed. So that automatically is put into the equation if you're using self lines. And this is for early generation trials using a formula, predicted gain formula developed by Brian Cullis. I can get into that in detail, but I'm sorry, there's no time today, maybe another day. Can work on genetic analysis and some important references, right? Do you have any questions? I want to show you one more trick which might be really useful in your actual analysis of field data using multi-site analysis. Do you have any? One more thing. Okay, some of you I know are very keen on using AceRemel. It's a really good algorithm, the AceRemel. We wanted to get it into DeltaGen, but we unfortunately could not, because then you would have had to pay for the program because there's IP on it, AceRemel. However, we just tried to short circuit the process and said, if you use AceRemel, you can enter, depending on your trial, what you have, years, seasons, locations, replicates, etc. You can enter the variance components, the outputs from AceRemel, together with the mean of the line blobs here, and then you have to depend. So if you are using in-red lines, early generation lines, you have to have the average of squared standard errors. So what I'm saying is you can add that, enter your AceRemel outputs, and then submit this information and go to your, it'll take you right to your breeder's equations for self-pollinated species, or half, whatever you want, depending on what you click here, right? So you have the option of bringing in AceRemel based data. All right. Any questions on what I have been boring you with up to now? Let's refresh. Go here. Kevin, can you hear me?
We don't have any questions.
Oh, that's
posted,
great.
So
that's good. It's good and bad. It doesn't matter. So what I'm going to look at here is a pattern analysis.
We got one question. Is there an alternative to using AceRemel?
The alternative, what do you mean? The alternative is just to get into DeltaGen and do the standard REML analysis. Okay, so let me say something. RemL is really good for large experiments where you have, large field trials where you have, generally you will expect trends. So it's very good for modeling and reducing that spatial variation in your large trials. And I would say, don't quote me on this. And that's one of the reasons why my former colleague, Brian Cullis, he developed, because he has been working on spatial analysis and nearest neighbor designs, etc., for a long, long, long time. And those of you who are wheat breeders, you know that even harvesting wheat, going up and down and up and down, it creates trends, right? So this was sort of developed for that, removing these trends and polishing up your overall bluffs that you get. Because as you start to reach a yield plateau, you're trying to pick up those tiny differences, make them significant. So this is very helpful. But again, it's up to you guys. If you think that you are the breeders, you know your environments, you know your fields, it's for you to make that decision, right? So I hope I answered the question.
Yes, thank you.
Okay. So what I want to do now is to do some multi-site. Okay. You don't, okay. So now all of you, I'm not going to, if you have the, you would have a file that I call performance plots, pattern analysis of multi-site data. Click on that and open it. Just tell me if you have opened it, Kevin, then I know that we are all on the same page.
Yep.
Okay. So if you look at the data set, if you go to blob metrics, right? So now when you run multi-site analysis, okay, you obviously, you want to often is to pick up what individual inbred lines or what individual synthetic varieties or how they, or even families, how they perform across your target population of environments that you sample. And then you also want to look at specific adaptation, right? Now, when you have large, so what you do is you analyze. So if in that particular matrix there, so Ballarat, Kerikeri, Kimahia, Palmerston North, Ruakura, and Waikato. So you have these different locations, and what I have done is analyze the performance of these lines in each of these. Are just names of cities. I've just put it in as a mock data set anyway. So I have analyzed each of these different locations separately, line performance in each location separately, and made sure that, how many lines do I have here? I think 75. And I have made sure that only locations where the line performance was significant were included in this matrix. If I had said another location, let's call it XYZ, and the performance of these lines at XYZ was not significantly different, I dropped it out. So what I'm going to use is only significant blobs, right? Data. So what I do here is, now I will copy that. Just copy it. Copy that. Go to DeltaGen and click on pattern analysis. I'm sorry. Oh, I'm getting a bit tired here. So go to that, go to the data input, clipboard, and paste your data here. And submit your data. So now you should see line names and the clusters. Don't worry about that. And this is the cluster number, but don't worry about that and that one. Okay. Okay. So sorry, can I get you to do it again? I'm really sorry. It's just that my head is getting tired. You leave the line names out and copy the cluster numbers only. Right, cluster analysis number. My apologies for that. Paste. Submit. There you are. Okay. So these are basically the line numbers. Then I go in here, go to line, and I click on cluster number and click run. Now the data is in the program. So you can see that these are lines, and then you have, these are the ways. So if you look at the summary up here, you see that line, only one factor, and all the others are traits, or in this case, locations, right? You can also do it for traits. Look at trait, how traits are related to each other. So then what I do is I go right to the top, press on pattern analysis, and you have, here you are, selected variables. So I want to look at all this, so I press that, and basically I include all these locations in my analysis. I just want to click on heat map if you wish, and then I say run. So what it has done now is analyzed or clustered all its data, drawn a dendrogram here, And it has given us a cluster, given a table of line groups. If I go here, if I point at any place and download the CSV file, let's take a bit of time here. Seems to have frozen. Should happen. Download. All right, let me go back here then. Just stay where you are. Let me refresh. I think I knew what happened. I was trying to do one analysis over the other. So if I go data input, clipboard, empty, paste, and paste. Submit data. Then go here, read quickly, cluster number, run. It's there, all there, pattern analysis. Right, put all these individuals in there. Run. Yeah, that's done that there. Okay. Let's see whether everything is there. Yep, everything is there. So it's not downloading for some reason. Why is that? To download table, that's what I have done. Sorry guys, for something. That's really funny. All right, let's see the download. I'll show the downloaded data in a minute. So if you go to cluster summary, what it gives you is a table that will give you the means. It doesn't download everything, the means here of each particular. Okay, so what I'll do is I'll go to this file. It's all there for you. So if you go to the pattern analysis part, right? That's what it does. It gives you the oh, what's happening here? Okay, please do bear with me for a minute. I will be back with you. Why isn't it downloading it? Copy this one. It does everything except download it. Okay, right. So it's not downloading it for some. See, all these means that you get here, basically should be downloaded, can download it, and you will get something like actually what you saw here. You will get something like that with the cluster number. It'll be clustered with a particular numbers. In this case, what you're looking at is if you go to the cluster here, it will tell you what cluster. Say line number 45 was in cluster 1, line number 51 was in cluster 1, line number 733 was in cluster 2, right? And it makes use of all this information that's here to generate a biplot, right? So in the biplot, generates a biplot, and so what we do here is in the biplot, I want to remove. You don't want to show those ellipses. Variable, you want your line number, which you can increase. So you can see your line numbers here. Variable size can be further increased. So these are your different sites where the locations where your trials were held, and these are your clusters, right? So you can see what cluster fitted into what group of locations. That is that shows you that. And if you were to go to cluster summary and go to this one, plot cluster means, so what it does is it plots your locations and cluster means. If you click on that, it gives you a plot like that. So what has it done? It has taken each location, taken the mean, that's the mean performance of growth at each location, and plotted it from the lowest to the highest. So we can call it environmental indices, from the lowest environmental index to the highest environmental index. Then it has taken each of the clusters, all the individuals that went into cluster 1, the mean performance of that cluster, it's plotted there. So you have, in this case, you have cluster 1, 2, 3. So in this case, it's showing only clusters. Maybe 3 was 3 shown here, and cluster 2. There are 2 clusters. Cluster 2 should be shown. There's something wrong here. So I'll go back. There's something wrong with the system today. I will show you exactly what it will give you. So if you go there, so this is what it will show you. This is what you saw, and that should what should come up here. There are 5 clusters here, and it's showing that this is the mean of each cluster within a particular location, sorted from lowest to the highest, and these dots give you a plot of the location means, so from the lowest to the highest, and then it shows you the relative performance of these clusters, different clusters at different locations, and enable you to look at the pattern of performance of these clusters across the locations, sorted from the lowest to the highest, right? So in this case, it's telling you that cluster 5 didn't perform very well at these low performing locations, but as time went on, it started to increase its performance. Now the question so cluster 1 was not good, and then the question is, what is causing these clusters, like these clusters, to not to perform very well at these locations, or what is causing these 2 locations, or even this one, to be the lowest? But also you find that a particular cluster here, cluster 3, performed above average, above the environmental index at the low performing, in the low environments. It may be the fact that it has some maybe resistance to nematodes or something like that, right? But as you get into other environments, it started to drop. So anyway, this gives you can have all sorts of discussions about that, but this gives you another graphical representation of line performance in terms of clusters across your locations, right? From the low performing location to the highest performing location. And of course, as breeders, you'll be interested to know what individuals were in these particular clusters, and that sort of will show you here. And later on, you can, you have the numbers of the individuals, you have their names, etc., and you can work backwards. So that's I think I have spoken a lot. I have given you a lot of information. Sorry if this didn't work at times. It's sort of, I don't know, I don't want to blame the internet and things, but these things do happen. I'd have loved to be in front of all you guys and take questions and discuss these things further. So as I told you in the morning, that it takes certainly more than 3 hours to run all the genetic analysis for self-pollinating crops and cross-pollinating crops, etc. So we can stop there if you want to.
Yeah, thank you. We just had a few questions.
Certainly, please do go for it.
Yep. In regards to the clusters, those were based off of BLUPs. Is that what those values were that we copied?
Exactly.
Okay.
Yep, they're BLUPs. So they're BLUPs taken from environments where their performance was significant. Where performance was significant, right? What I would advise you to do is to, if you go to the multi-trait one and open there, you'll have another matrix there. Maybe I'll just do it anyway. If you bear with me for 2 minutes, go to DeltaGen. So what I've done, I've taken the matrix there. So that's a genotype by trait matrix. I go to input, paste the data in there. You'll see how that works. Paste. And so remember, these are all BLUPs, right? So these are all BLUPs for the different traits. Okay. And also when you do pattern analysis, for these location or traits, they're environmentally standardized to bring them to an equal scale. So in this case here, and that's very important when you actually, and they're standardized to make sure even looking at locations, is that the high yielding locations do not have a huge impact on the overall picture. So they're all scaled. So these are traits. So if that is very useful in pattern analysis. You can look at how well they're related. See, let's just take those traits. So I say standardized data. So because these traits, for example, they could be measured at using different scales, some in grams, some in kilograms, some in millimeters, right? So they're all standardized within each trait, so that you're comparing apples with apples. So if you go to cluster analysis and we run it, so there you are. So let's see whether it works this time. Download. Yep, it worked. There must have been something wrong in the data set that I had to check it. So, so there you are. So that gives you your clusters. So if you go to these clusters here, you can't see what I'm showing you. Yeah, I will stop sharing here and I will share. Where is this? I believe it is this one. Oh, is it this one? Data. Right, I think that's the one. Can you see it now? Yeah, so that's the output that came out from that analysis. So if you go to clusters, I'll do it a bit fast. I'm sorry, just to make sure that you get what I'm talking about, and you can sort it by cluster. What happened there?
Sorted by line.
Thank you. By cluster. There you are. So all the members of cluster 1, all the members of cluster 2, right? So all that information is got very easily. Okay. So I'm going to stop sharing and I'm going to share it again with DeltaGen. Go back here, share. Right. So that is what you saw, right? Red transferred. And then you also get a cluster summary if you want to look more on what sort of variation do you have within clusters. So it's telling you, say, cluster 1. This is the mean for this trait in cluster 1, mean for that trait in cluster 1. Then you have the standard deviation. Right, starts from here. Sorry, starts from here. Standard deviation for dry weight, standard deviation for leaf size, standard deviation for plant height, stolon density, stolon number, I think root shoot ratio or something like that. So it gives you sort of what variation do you have in the clusters. Then if you go drawing performance plots, this makes sense because they're not, it doesn't make sense. So if you go to that by plot, so what it does is I just remove we wanted to have, some people like to have this arrow ellipses. So we just did it. So we go to variable, we want lines. We increase your lines here. We can, your variables. So you see that, right? So these particular traits, the correlation, you can see the correlation there, and you can see how these different clusters contain individuals that have high expression, low expression for these traits, so you can do a lot, that sort of thing. Now, you can go into things like, I don't have time to go into the more detailed multivariate analysis, but like you can do MANOVA, you can do selection indices, there's no time. But if you were to do this, for example, all that, it also draws a very crude diagram. And if you cut that, it gives you the Pearson correlation coefficients, right? Based on blobs, the blob ones, okay? So it gives you, this is standards R stuff. Those of you are familiar with R. So it gives you that. Now, in terms of the question, yes, the first question, yes. Whether you're using locations, genotype by location matrix, or genotype by trait matrix, it's up to you. You can use blues if you want to, if you do use a fixed effects type of analysis. But since I'm a plant breeder, I prefer to use breeding values, or which are blobs. You can call them genetic worth of a line or individual. So I prefer to use that. Okay. So if you use MANOVA, you can you basically you write your equation here and run it. Then you have your weights and things like that. You can add, not for this, but for using the. So that's maybe another day. If you guys want to see me again after today. But yeah, so go to there were some other questions you asked me.
Yeah, somebody asked about two-step analysis, as in doing your single location adjustments and then across locations.
You can do that. Yeah. It's up to you. So you do it in a single location, and then, yeah, I always advise people to do two-step analysis, because when as a breeder, you want to say, right, how did they perform at each environment, in each environment? Were they significant or not? And then, you don't have to do multi-site location analysis, two-step, but it's again up to the questions you're asking in your breeding program, what your objectives are.
Now, would you want to do that, or can you do that with, you have to do each individual location and upload the adjusted values across locations?
Yep. Yeah. So, okay. So if you're doing a single location analysis, right, there are two things here. A single location analysis, you analyze each data set collected from a particular location individually, right? And you get to individual blobs. And you want to do something like pattern analysis to summarize, right, what you want to do. Then you take those blobs as a column from each particular location. It's location A, B, C, D, E, F, G, H, I, J, K, and you put them and you make that, develop the matrix, the two-way matrix, right? And then you take that matrix of blobs or blues, depending on what you want to do, and then upload it into DeltaGen and you do your pattern analysis. Okay. Now, when you want to do a multi-location analysis, what you want to do is you want to find, your question you're asking generally is, how did these lines perform on average across the population of environments that you have sampled? Okay, so you're going to get a single column of blobs. Okay, less like the mean across all environments, right? Now, to be able to do that, but for DeltaGen, you just stack one over the other. So you have environment A, you get your raw data, and under that you you put your environment B raw data, the normal stuff, and then feed that data into your spreadsheet or into your data matrix. So if you go and look at the data set that I've sent you, say for example, data set D, matrix across sites, asterisk growth. So there you can see how I have across, perennial ryegrass. So that you can, this is across sites with an asterisk. So you can see that how I have stacked them, one location over the other.
We're seeing your DeltaGen.
No, that's fine. But you have
Oh,
the, you can, if you just click that open, if you look at D,
Yep. I see.
Yep. Okay. So, yeah. So, hope I answered your question.
Yeah, I think
any other questions? Any other questions?
I do not see any others in the chat or the QA. So, please post them.
All right.
Yep.
So, thanks, guys.
Thank you. This is fantastic. So, appreciate your time.
No, it's all right. It's a pleasure, and wish all you guys, all the students, especially, the very best in your studies. And hi to my mate Valerio. And we'll catch up sometime. All right.
Yep. So yep. You said you'll be willing to share the software with us?
I will.
Disperse it to the
attendees.
If
if one of you send me a Dropbox, I'll put it into that and send it off to you. Okay.
Okay. Thank you.
All right. And all the very best for Christmas and the New Year.
You too. Thank you.
Okay. See you guys. Cheers. Bye.